Hello people, now we are solving some questions on the topic called prokaryotic cells. So the first question is, how many of the following represent prokaryotic cells? The first question asks that how many of the following represents the prokaryotic cells? Bacteria, blue-green algae, mycoplasma, PPLO. So how many of the following represent prokaryotic cells? Bacteria, blue-green algae, mycoplasma, PPLO. All four of them are going to be represent the prokaryotic cells. Next question is statement based question. The statement 1 and the statement 2. The first statement says that prokaryotic cells are generally larger than eukaryotes. Second statement is prokaryotic cell multiplies slower than the eukaryotes. The first statement is the prokaryotic cells are generally larger. This is a wrong statement because the prokaryotes are generally smaller than that of eukaryotes, and so the first statement is wrong statement. Second statement is prokaryotic cells multiply slower than the eukaryotes. This is also wrong statement because they multiply at the faster rate, and the statement 1 as well as statement 2, both statements are a false statement. Next is the match the following. First is, so the match the following given on the basis of their shape. If you want to match them on the basis of the shape, if you want to match them. So vibrio means comma shaped bacteria are called vibrio, and spirillum, spherical shaped bacteria, spiral shaped bacteria are called as a spirillum. Bacillus, rod shaped bacteria are called as bacillus, and coccus means spherical. This is the match the following, and they are given certain kind of statement. How many statements are correct statement? The first statement is the organization of prokaryotic cell is fundamentally similar even though the prokaryotic exhibit a wide variety of shapes and functions. First statement is a correct statement. There are 5 basic shapes of bacteria. This is a wrong statement because there are only 4 basic shape present in the bacteria, so this statement is a wrong statement. The outer boundary of all the prokaryotic cell is cell wall. This is a wrong statement because the mycoplasma which is belongs to the prokaryotic do not have any kind of cell wall. So here they have mentioned all the prokaryotes, so hence this is a wrong statement. Fourth statement is prokaryotic have well-defined nucleus. This is also wrong statement. The prokaryotes do not have well-defined nucleus. So the correct statement, the first statement is the correct statement, and rest 3 are the wrong statement. Hence, only one statement is a correct statement among the following. And the next question is assertion and reason type of question. The first assertion is the genetic material of prokaryotic cell is basically naked. Yes, assertion is true. And the nucleus of this cell is not developed. This cell means here they are mentioning about prokaryotic cell. The nucleus of this cell is not developed by nuclear membrane. The reason is also true. So once again, whether the reason is correct explanation of assertion or not, let us see. The genetic material of prokaryotic cell is basically naked because the nucleus of these cell is not developed by nuclear membrane. Yes, assertion is also true and the reason is also true, and the reasons correct explains the assertion. The reason correct explains the assertion. This is your answer. And the next question is which of the following confers unique phenotypic character to the bacteria? So plastic, wrong statement. Nucleus is also wrong, and both A and B this is wrong. Answer is plasmid. So the plasmid is going to be provide a special phenotypic character to all bacteria. Next question is which of the following is unique character of prokaryotes. The ribosomes, they are present in both eukaryotic as well as prokaryotic. Endoplasmic reticulum is present in the eukaryotes. Lysosome present in the eukaryotes. So the answer is mesosome. So the option is B. These are the unique character of all type of prokaryotes. Next is match the following. We should match this on the basis of their size. On the basis of their size, we should match. Typical eukaryotic cells are 10 to 20 micrometer in the size, 10 to 20 micrometer in the size, and the typical bacteria is 1 to 2 micrometer within the size. And the PPLO is all about a fluoropneumonia like organism. It is about 0.1 micrometer within the size, and the viruses are 0.02, say like 0.2 within the size. And the next question is, how many statements are incorrect statement? We should find out the incorrect statements. First statement is, the bacterial cells have chemically simple cell envelope. Wrong statement. Within the Ncert, they have mentioned that the bacterial cells have chemically complex cell envelope, not simple. They are chemically complex envelope. Hence the first statement is a wrong statement. And the second statement is, cell envelope consists of loosely bound 3-layered structure. This is also wrong statement, because it is not loosely bound, it is tightly bound. It is not loosely bound, it is a tightly bound structure. The third statement is, ribosomes present in both eukaryotics as well as prokaryotes. Correct statement. So hence the ribosome is also known as the universal organelle. Universal organelle, because it is present in both prokaryotics as well as eukaryotes. Last statement is, mesophyll cells are found round and oval in the shape. This is a true statement. So two statements are wrong statement and true statement are correct statement. Hence the incorrect statements are two among the following. And the last question is, arrange the following layers of cell envelope from outer to the inner side. The outermost layer is something called as the glycolax. This is the first outermost, and the second is cell wall. This is inner to that of glycolax, and innermost of all of this following is cell membrane. This will become third. So the outermost is glycolax, and within the glycolax there is a cell wall, and within the cell wall there is a cell membrane. So these are the sum of the questions, and thank you, and we will see in the next session.